Ayan, good day. Welcome to the Accessible Intervention Material in Science. So, I am JRAEugenio. So, let us start our intervention learning. So, our topic will be about DNA and its structure. So, before we start, let us observe this picture. So we have here different fur coats of horses. So we have here chestnut, palomino, and cremello. So next we have different combs of chicken. So looking at the left side here, we can see that there's a walnut, pea, rose, and single. And on the right side, you can see some other combs of chicken, different varieties. And also in humans, we have pattern baldness on the left side and no baldness at all on the right side. Also, we have these three cute puppies showing different fur coats of Labrador Retriever. So we have brown or chocolate, golden or yellow, and black coat colors. And also we have different earlobes such as the attached and pre-earlobes. So we have a question here. How do these traits appear? So there must be an explanation for that, and let's see. So those traits came or being dictated by the DNA. And your DNA and our DNA exists as a molecule inside the nucleus of a cell. So, the sperm cell and the egg cell of a mother and a father will join and they will form a zygote combining all the genetic material or the DNA from the parents. So, you will develop some traits from them. Okay? So, here are the two different kinds of nucleic acids that can be found in our body. So we have the DNA or the deoxyribonucleic acid and the ribonucleic acid or the RNA. So there is a difference between the two. So as we go on, you can later learn the difference between them. So here is a chemical structure of a DNA. So going back a while ago, you can see here that the DNA has two strands and the RNA has a single strand. Let's pick a single strand of a DNA. So as you can see here, there is a repeating molecule here, and we call that as the nucleotide. So a nucleotide is a compound, which is a combination of three smaller compounds, which are the phosphate group, sugar, and the nitrogenous base. So let us differentiate the three smaller molecules and compare them between the DNA and RNA. So in the phosphate group, you can see that, have you seen any difference? There is none. There are no difference between the RNA and DNA. How about in the sugar? Looking at the sugar, there is a slight difference between the RNA and DNA. And the sugar of ribose or ribonucleic acid is called as ribose, and the sugar of DNA is called as deoxyribose. Now, the difference here is that the ribose has an oxygen atom at carbon number 2. But comparing it to the deoxyribose, only the hydrogen atom is the one attached or chemically bonded to the carbon number 2 of this sugar. So that is the only difference. So that's the reason why is it called deoxyribose, because in Latin when you say de, it is to remove something, and what has been removed? It is oxygen or oxy. So that's why it is called deoxyribose. Now, how about the nitrogenous base? So the nitrogenous base, we have two kinds. So the one with the single ring or chemical ring is also known as the pyrimidine. And the one with two rings is also known as the purine. So let us now compare the different nitrogenous bases in DNA and RNA. So in RNA, we have here four nitrogenous bases. We have cytosine, uracil, guanine, and adenine. While in DNA, we have cytosine, thymine, guanine, and adenine. The one that is, let's say, the one that is different between the two is the appearance of uracil in RNA and the one or the appearance of thymine in DNA. So the other three nitrogenous bases exist in those two nucleic acids such as the cytosine, guanine, and adenine. So let's move on. The nitrogenous bases are also paired up or they have base pairs. So in RNA and DNA, guanine and cytosine can be paired up, while in RNA, adenine is paired to uracil, while adenine in DNA is paired to thymine. So that is the difference between the two. Now, since we now know the components of the nucleotide, let us now create a, let's say, a strand of DNA. So when you combine different nucleotides of a strand, you can form a single strand, and we have here two strands. Since DNA has two strands, each has a single name. So the one on the left side is also called as the leading strand or the 5 to 3 strand, while on the right side, it is called as the lagging strand or the 3 to 5 strand. Now, why is it called the leading or 5 to 3 and why is it called lagging or 3 to 5? The reason why the left strand is also called as the 5 to 3 strand because let us go back to the carbon atom. The carbon atom in the sugar has been named from 1 to 5. So the topmost carbon atom here is number 5 and the bottom here or the most bottom carbon atom is 3, neglecting the number 2 because the topmost is the 5 and the most bottom is the 3. So that is why it is called 5 to 3 strand. Now, look at the other strand. So I think that the nucleotides has been flipped, and that is the reason it is called 3 to 5 because the topmost carbon atom now is carbon atom number 3. Okay? So the most bottom now is the carbon atom number 5. So that is why it is called 3 to 5. Now, the two strands must pair up so that it can form a DNA. So as you can see here in the middle, the nitrogenous bases has been paired up, adenine and thymine, guanine, cytosine, adenine, and thymine. So another further a diagram that shows the bonding between the two strands. Okay? So I hope you understand now the DNA and its structure. Thank you for watching.